Good morning and welcome back to the Solutions Showcase stage. I'm Jenny Stevens. I'm the Director of Channel Strategy and Implementation at IFT First. So our next session takes us into nanoscale and shows us how a tiny innovation can have a huge impact on food and beverage performance. Before we get started, I do want to remind you that at the end of the session, please scan the QR codes on the back table with your IFT First app for a chance to win prizes. So this session, Ultrafine Bubbles as a platform for functional and sustainable food innovation, sponsored by Hydrosome Labs, explores the power of ultrafine bubbles, also known as nanobubbles. These nanoscale gas cavities can enhance mouthfeel, nutrient delivery, and even fermentation efficiency, helping brands improve functionality, speed up production, and support cleaner labels. Here to tell us more about the science behind nanobubbles and how they're being applied is Nick Jackowetz, Chief Scientific Officer at Hydrosome Labs. So please join me in welcoming him.
Thanks so much. Awesome. Hi, everybody. I'm Nick, as was mentioned. I'm the Chief Scientist at Hydrosome. Really happy to be here today to talk to you about some of our technology in the ultrafine bubble, nanobubble space. It's a really cool, really new, innovative idea in nanotechnology, and really happy to talk to you guys today about the applications that are pertinent to food science and fermentation. So let's get started. See if we can advance. There we go. Cool. Great. So today we're going to talk about a lot of things. We're going to talk about the science of nanobubbles. These bubbles are very small. They're very little. They seem like they might have no impact at all. Many of us haven't even heard of a nanobubble or an ultrafine bubble, but they're really cool, really innovative bubbles that we're playing with now and learning a lot more about in the scientific field. We'll talk about their performance. What can they do? How can they speed up or change chemical reactions, biology, microbiology, fermentations? What application areas are really interesting that we're seeing for using nanobubbles today? How are they important in food and beverage? And then how do we implement this? How easy is it to use ultrafine bubble technology, and where is it being used today? And then finally, this is a really emerging science. So right now, we're one of probably the few nanobubble companies operating in the US, and we're always open to collaboration, new opportunities to explore new application areas for this really fascinating technology. So today, when we think about the nanobubble space, it's really confined to some traditional areas, which is wastewater treatment is a really classical area where you'll see this be used currently. You'll also see it in agriculture. You'll see it in cleaning. So these bubbles have been used there for a little while. But we're really branching out and pushing it into other spaces like fermentation and the CPG space. So right now, we're commercializing cosmetics, using it in topicals to enhance permeability of active ingredients. We're pushing more into the food and beverage space for texture, emulsions. And then fermentation, we'll talk about how we're using them to boost efficiency and overall fermentation yield. So it's a really exciting time for nanobubbles. If we think about, though, some of the gaps in the spaces that we're looking at in CPG and fermentation, they're common ones that we know. We're talking about the limited availability, bioavailability of ingredients. We're talking about a lack of innovation in sensory and texture. And always there's a quest for cleaner, sustainable ingredients in the space. Likewise, in fermentation, we want to have faster, more robust fermentations. We want higher titers. We want higher yields. And these are issues that we already encounter today. So this is where nanobubbles can help, and this is where we play. Great, so how does this all come together? So at Hydrosome, we have a patented technology that's able to infuse nanobubbles into liquid streams. So this could be pure water. This could be a fermentation with cells in it. This could be dairy, milk. This could be a finished beverage. This could be a process. It could be lots of different things where you have an aqueous solution where we can impart gases and make ultrafine bubbles. So we have a patent on that where we can make very specifically sized bubbles, and we can alter that concentration, and we can alter the gas that's in them as well. And what this does is it creates an environment where we can manipulate microbiology, so how cells react to these bubbles, how they grow, and also chemical reactions too, how fast these chemical reactions happen, and other kind of physical chemistry reactions as well. But let's talk about bubbles for 1 second too. The bubbles that we all know and encounter day to day in our sparkling water, beer, champagne, these are coarse macro bubbles. These are large bubbles that we see. They're transient, which means that they are buoyant. They will leave solution over time. The bubbles that we're talking about here are ultrafine bubbles. They were only defined by ISO in 2017. This is still a very new area, and these bubbles are much, much smaller. And what happens when you get much smaller is these bubbles and how they're governed totally changes. So now imagine a bubble that's so small that it doesn't float anymore. So a bubble that stays in solution for weeks, months, if not years at a time. So that's really interesting and changes how we think about bubbles overall versus these bubbles that just leave solution very quickly. So again, the standard for bubbles, ultrafine bubbles, is 1,000 nanometers. Our hydrosomes are about 100 nanometers. So to put that in perspective, that's about the size of a virus particle, something very, very, very tiny. Another way to think about it is if you have a champagne bubble 1 millimeter in diameter, you can fit 1 trillion of our ultrafine bubbles inside of that champagne bubble. So just crazy scale, very, very small. And that's really the essence of this technology. It's smallness and the surface area that it brings. So if we look back in history of this whole area, ultrafine bubbles exist in nature. It's not something new that we've developed or we've commercialized for the first time. These have always existed. They've been present in lakes, streams, rivers, waterfalls over time. It's just that the concentrations that you find them in these areas is quite small. At Hydrosome, we've harnessed these bubbles for high concentrations, for their size, and for the type of gas that's inside of them for different applications. So it's always existed, these bubbles. We're just really harnessing them in these new and exciting ways. And the science, as you can see, only started in the 80s and is now like pushing forward as our understanding has gotten much better about them. So ultrafine bubble technology is really interesting too, and some of the principles of these bubbles are fascinating. If we think again about these bubbles, they're so small they don't float. So that's a really weird concept to think about. We can think about particles, we can think about suspended solids, but a bubble that doesn't float is an inherently strange idea. But that's what happens when they're so small. They're not governed by buoyancy anymore, they're governed by Brownian motion when they're that small. The physics change. Also, we can think about these bubbles as really effective delivery agents. So the bubble itself is coated with a layer of hydroxide ions, OH-. So naturally, when the bubbles are in solution, they have a negative charge, a negative zeta potential that we can measure. So the bubbles can interact electrostatically, so they can interact with things based on charge. They can also interact with things based on hydrophobicity. There's a hydrophobic layer at the interface between the gas and the liquid too. So there's a lot of ways these bubbles can interact. And then lastly, the gas payload is really interesting. As we get smaller and these bubbles shrink, what's called Laplace pressure, or the pressure inside the bubble, increases dramatically. So a bubble that's 100 nanometers has a theoretical pressure of 30 atmospheres. So its gas carrying capacity, its gas payload, is very, very high. And that's really interesting and has a lot of implications, say, in fermentation, if we're trying to feed an organism that wants more oxygen, for example. So a lot of really interesting properties. And if we think about, again, this technology and other types of people that are playing with it, other competitors, there really isn't many. Again, we're one of the first in this space to push ultrafine bubble technology in the US, certainly. It's existed in Asia and Japan in particular for a number of years, but really not to this level and not in these application areas. So we're really changing it and customizing these bubbles, changing the size, the concentration, the type of gas that's inside of them. That can have unique application areas. We're looking at obviously making it sterilizable so it's clean. It works in food areas and food systems and in food processing plants. It's very energy efficient. To operate one of our units is about $10 a day, so it doesn't take a lot of energy. It's highly sustainable, and it's very simple to operate. Very easy to use, a very simple interface. If you want to look at these bubbles, it's quite easy, actually. If you have a laser pointer and you have ultrafine bubbles in a solution, a clear solution, you can visualize them. And so you can see that here on the screen where we look at cuvettes and we increase the concentration by about an order of magnitude each jump. And you can see that intensity of light scattering increases as we keep going from left to right. And so that's a really easy way. We can QC check them that way. We can make the bubbles and just double check that they're there. But if we want to dive deeper and really make sure that we're producing the same quantity, the same size of bubbles, we can use something called nanoparticle tracking analysis. This is similar, kind of a sister type unit to dynamic light scattering, DLS. So this piece of equipment, I'll show you here, is a flow cell where we can put in liquids. We can look at how these particles move, and based on how fast they move, we know their size, and then the microscope picks up how many are on the screen, and we can count them. So here's a deionized water that we put in there. Not much going on. You see not many red dots, nothing else happening in there. But if we now change it to the ultrafine bubble solution, we have a different story that emerges. So once it slows down, you'll be able to see All these little guys floating around are ultrafine bubbles. Both of these samples are optically clear. You can't see anything with the naked eye, but once we apply this type of technique, NTA, we can measure these bubbles. This takes about 2 minutes. So we can just put this into the flow cell, we can see how many there are, we can see the size and concentration, and then go from there. So do a QC check and make sure everything is looking good. So this is the standard for measuring bubbles. We have it in our lab, and it's a very common unit. Implementation of using this technology can vary. And so in fermentation, for example, it's a really simple process. We have our ultrafine bubble generator, and we can park it, put it right next to a fermentation tank. So we can sit right next to that fermentation tank, and we can essentially do a pump over or recirculation. So imagine we have a tank, you're fermenting something in there. We take a stream out of the bottom of that tank, we pull it through our unit, we infuse ultrafine bubbles inside that unit. We then return that to the tank. Now it's infused with all these ultrafine bubbles, and we keep doing this repeatedly. We do this because we want to keep having more bubbles in solution, more surface area. Also, the cells are dividing. We want more bubbles to compete with all those cells that are now growing during that process. So we found this to be the most effective. Having more bubbles during the fermentation really helps, again, with that mass transfer, moving gas and solutes to cells more effectively. And what we see in fermentation is really an efficiency play. We're able to really decrease the time it takes to complete a fermentation. We have higher titers, higher yields, and what's called better oxygen uptake rates. So a really effective mass transfer of oxygen. In a lot of aerobic fermentations, moving oxygen to a cell is often limiting. If we can improve that, like we've shown, we can really boost the potential of a lot of fermentations and increase their outputs. So that's a really important note. How does this all work? How does it all come together? There's a concept that we call attract and deliver, where you have a lot of bubbles, a lot of surface area. They're interacting with solutes. They have a gas payload in them themselves. They can stick things to their surface. They can bring gas closer to cells, and then they can bring those cells, those solutes, or that gas, and really boost its output and boost its growth, bringing cells the things that they need. We've done even work looking at genetic sequencing and looking at gene expression, where we can expose a cell to ultrafine bubbles and take a look at how its gene expression changes. And there's an important few things that we notice, especially as it relates to oxygen. So as we expose these cells to oxygen, they upregulate genes that need oxygen. And so we see an upregulation of the citric acid cycle and also oxidative phosphorylation. So really helping push energy potential for these cells. At the same time, transport mechanisms are also highly upregulated. So we see a lot of movement across these cells as well. So we know, based on our hypothesis, that this seems to make sense. We're moving gases and solutes more effectively to these cells. And that means when we're talking about where does this work best, highly aerobic fermentations and also fermentations with a high biomass or high cell count. We can really help deliver into environments where they may be lacking, where there may be a lot of competition for nutrients and gases. This is where these little bubbles can really make a difference. And so we've done a lot of studies with various groups, clients, universities. One of our best partners is the University of Illinois at Urbana-Champaign, where they have the IBRL lab. And down there, we have access to tanks that are up to 1,200 liters. And so here's an example of a fermentation we did with them at the 400-liter scale. So it was a fed batch fermentation with E. coli. The only difference was the infusion of ultrafine bubbles in one treatment versus the other. And we were looking at outputs like growth, doubling time, and other types of metabolites. And when we look at this, we can see some differences here for this one example where the ultrafine bubble hydrosome treatment in the light blue line really takes off more quickly. So it starts to hit that exponential phase much more quickly versus the control, which doesn't have the ultrafine bubbles. Also, the biomass that we achieved in this was lower in the control versus the hydrosome. So we were seeing some benefits there too. When we dove deeper, we saw a doubling time that was about twice as fast for the hydrosome treatment, so twice as fast doubling time. And also what was really interesting was how are these cells using the sugar that it had? What is the mass balance? Where is this carbon going? And we noticed with the hydrosome, a lot more of that carbon was going into cells and not going into other secondary metabolites or CO2, and that was really important. So we saw that. We also looked at things like oxygen uptake rates, carbon dioxide emission rates. They were higher in hydrosome. Again, higher respiration, higher growth potential in these cells, better efficiency, better outputs at the end of the day. And then as well, we looked at secondary metabolites too. That's important to look at. Again, where is that carbon going? And we saw in the hydrosome treatment, a lot less carbon was going to secondary metabolites. Again, it was going into biomass and where it was needed. And we weren't seeing as many things like acetate or ethanol, which are also indicative of fermentations that are lacking oxygen. So we're seeing here better movement of carbon and less oxidative byproducts overall, which was really encouraging. We've also done some work on the Saccharomyces side, so with yeast too, not just thinking about E. coli, but how can we affect yeast growth and fermentation? So we have a lot of clients that grow yeast and want to have more biomass with yeast cells. So we did an experiment looking again, infusing ultrafine bubbles in one case and no ultrafine bubbles in another case in this batch fermentation, and looking at the growth of these yeast cells. And what we could see is, if we know yeast, we know how they grow. If we give them a lot of sugar, they're going to be anaerobic, right? They're going to ferment. That's called the Crabtree effect. So once we get beyond all that sugar, and once that sugar is metabolized, now we enter an aerobic phase, which you can see delineated by the line in the graph. So you can see when it's anaerobic, we're seeing a little bit of difference between the hydrosome, which is a light blue, and the dark blue, the control, but not a ton of difference. But as we go into that aerobic phase, that's when you really see the hydrosome technology and the ultrafine bubble technology shine, and we see a differentiation between the two treatments. So we have a much higher level of biomass produced when we have these ultrafine bubbles in solution by about 60% versus the control. So again, we're not seeing that control be able to reutilize the carbon source, which is ethanol in this case, and turn it into more cells. So this was pretty big, and this is something that we're working on with a lot of yeast producers at the moment to increase their biomass outputs. So fermentation is a big area for us too. We just covered a lot in that space. Really important. This is only the tip of the iceberg. We have a lot more data in this space, but suffice to say, it's really interesting, and there's a lot in this space to explore. But we're at IFT after all, and I think there's a lot of really interesting food applications here too to talk about and mention. We're seeing really interesting changes and benefits of the bubbles in things like beverages, where we can add the bubbles at different concentrations or with different types of gases and get a different mouthfeel, get a different expression of how flavors taste in the presence of ultrafine bubbles. So there's some really interesting innovation that can happen here too. And again, these are just bubbles. There's no surfactants here. It's a clean label idea. It's just something that we've designed and made in a very finely tuned, engineered way. Also, a big area for us is emulsions. So how do we get oil and water to mix? How do we sustain these emulsions, things like mayonnaise, things like flavor emulsions? We're noticing that the bubbles can actually help and stabilize and actually enhance emulsified systems too. So there's some really interesting work we're doing with some partners in this space. In the past, we've done work looking at delivery of active ingredients, things like zinc and iron, into the gut more effectively. So we've done some bioavailability work there. And then the microbiome, we're in the midst right now of submitting a paper for publication looking at the effects of ultrafine bubbles on the gut microbiome and other health markers. We'll talk about that too today. So there's a plethora of application areas here. We're just scratching the surface of what this technology can do. But let's start here first with a kind of a classic example of oil and water. So here we have two water samples. We have the hydrosome with the bubbles in it on the left, and we have the control on the right. This was just the water, and then we put in some canola oil on top of it, and then we took just a simple immersion blender like you'd have in your kitchen, and we mixed it for 10 seconds. And then we did was we took the immersion blender back out, and we waited over time to see how that oil slick would emerge. And if we watch here, this is over about an hour-and-a-half period. You can see the control, those bubbles really start to come back and agglomerate much, much more quickly than the hydrosome case. So we're able to maintain those small oil droplets just with these ultrafine bubbles alone, right? So you can see how all of those oil globules come together in the control, whereas the hydrosome, they stay apart. And we think that's because these ultrafine bubbles are acting like something called Pickering reagents, where they're able to interact and get in between the oil droplets, preventing further agglomeration. So this has really interesting implications in the emulsification category, being able to use these bubbles in conjunction with emulsifiers, potentially even reducing the amount of emulsifiers needed, reducing costs, reducing off-taste by those emulsifiers as well. So it's a really surprising finding and really interesting area that we're pursuing further. We've also looked at things like common ingredients like mayonnaise, which is an oil and water emulsion too, and found something similar where we can modify the texture and the sort of flavor and smoothness of mayonnaise here. So we have 2 mayonnaises, one on the left, one on the right. The Hydrosome's on the right. We're seeing a bit more thicker mayonnaise production there, a thicker feel, a thicker viscosity, smoother mouthfeel, less graininess, less coarseness in this. This was an example to show some of the texture a bit better. We have other ones too where we've done them with a higher shear, so we have a whiter mayonnaise, but you get the idea that with only a 5% addition of ultrafine bubbles here, we can change the texture, and that's a big one too. So a lot of these food systems where texture becomes important, we don't have to think about adding gums or other things like that. We can think about adding these bubbles to modify the texture as well. And then similarly, we work, as I mentioned, in the cosmetic space. So there's a lot of work we've been doing there, and people always want these cosmetics to last longer. So we've done accelerated studies with cosmetics and shown we can increase the shelf life of a cosmetic ingredient by about 2-fold. So we can really prevent its separation over time, make that ingredient have a nicer feel, a nicer absorption into the skin, and have that cosmetic have a longer shelf life where it can be sold for longer periods of time. If we dive into that cosmetic a little bit further and look under the microscope, I think you can see it here too. So on the right-hand side, we have the hydrosome, on the left-hand side, the control. Look at the size of those oil droplets in the hydrosome. So we're able to really make a smoother and finer emulsion of those oil droplets versus the control with the same emulsification system. The only difference here is little bubbles that are in there, really helping to keep that emulsion nice and tight, nice and small. And what this means for our clients at the end of the day is that we have a superior product with better performance. It's less greasy, it's less sticky, it's less tacky. It goes into the skin, absorbs better because you have these smaller droplets in there versus these larger globules of oil. So it's a really interesting finding. So it extends beyond food, and in this case cosmetics, but it's a really important application area for these bubbles. And then I'll say lastly, one of the areas that I mentioned before is the microbiome. This gets a lot of attention as of late. There's a lot of really interesting health implications in the microbiome, a lot of regulatory hurdles to think about in the microbiome. We were seeing research out of Asia showing that with a supplementation of water containing ultrafine bubbles, there was significant modifications to the gut microbiome. And that made us think, okay, we're making our own style of ultrafine bubbles. We're filling ours with air. Let's see what happens, in that case, because in these studies, they were filling them with hydrogen or nitrogen and seeing meaningful differences. But let's see how our technology stacks up too. So we funded a preclinical study with lab rats. So we had 24 rodents, 12 in each group. And one group had the ultrafine bubble water as their water of choice. That was what they would drink, and they could drink that as they wanted to. It wasn't fed by gavage. They could do it at libitum. So they could drink it whenever they wanted. And the other group just had regular water. And then we compared over time health markers across all kinds of things, inflammatory markers, short-chain fatty acids, histopathology, all those kinds of things. We also looked at a modification of the concentration of the bubbles halfway through too to see if there was a dose-response effect. And so first, one of the interesting things we saw is the short-chain fatty acid concentration with the ultrafine bubbles was significantly higher with the hydrosome. So this is an important marker because short-chain fatty acids in the gut are an indicator of health. The more you have, the more they can help feed other bacteria that are in the gut. They also feed the cells on the lining of the intestine. It keeps a healthier barrier there too. So after 8 and 12 weeks, we had significantly higher levels of those short-chain fatty acids in the gut, which bodes well for the bubbles doing something there. And then again, looking at things like inflammatory markers, after 12 weeks, significant reductions across all 3 inflammatory markers that we tested with the hydrosome treatment versus the control. So much higher inflammation in the control, much less in the hydrosome over that time. So again, important. It speaks to the bubbles. It speaks to dose response too, because again, after 6 weeks, we increased the amount of bubbles in the diet by about 3 orders of magnitude, and this is when we really saw effects of these bubbles. So we know the more bubbles in this case, we had a more pronounced effect of these findings. And then lastly, we noticed, again, different compositional shifts in the type of bacteria that are in the gut. Obviously, the gut microbiome is a very complex system, but the main takeaway here is we saw differences in the kingdoms and shifts over time, where we had a higher concentration of a kingdom called Bacteroidetes and a smaller a drop-off in the Fermentacuides too, which is generally a positive thing and, as kind of prescribed by the literature. And so overall, we're seeing a lot of good benefits here. Again, the main takeaways are going to be around higher short-chain fatty acids, lower inflammation, no other pathologies noted using the bubbles, so no adverse effects found here. And this is just supplementing with water with these bubbles over a 12-week period. So everything else being equal, these bubbles have an effect. And so one of the main things to kind of wrap up with here is this technology, it has so many interesting and broad applications. We're only scratching the surface at where this technology can go. There's a lot of customization that can happen. It's new, it's novel, it's untapped, it's sustainable. And we're seeing a lot of interest from a lot of large and small food companies alike looking at this technology with a closer lens because it is truly fascinating. Doing all this with just a bubble and having all these impacts is truly remarkable. So we're seeing these great benefits. We're seeing efficiency gains, increases in production, and lower costs overall. So it's really an important, exciting area. And the main takeaway, excuse me, think about is really transforming water from a filler to a functional ingredient. So thinking about water in new ways that you haven't thought of before, using it to just boost and change textures, change taste, change the reaction kinetics of your system, all with just adding ultrafine bubbles. So we're really excited about this technology. We're Hydrosome Labs, so we have a booth in the startup zone too, so feel free to stop by and talk to us afterwards as well. So we're located in the startup zone. We'll be here as well too, but again, really excited about this technology, and we welcome any questions you may have. So appreciate the time. Thank you very much. Okay.
Questions? Questions?
Hi, thank you so much for the presentation. I had two questions. So how does it work on a benchtop level with the Hydrosome bubbles? And have you done any research or food studies to see how the taste impacts on consumers? And is it a positive kind of impact?
Yeah, great question. So on the benchtop side too, we have a unit right now that we're in the process of making that is much smaller. Right now we have larger ones for fermentation, which is where we started, but we do have benchtop units available too. I will say we can also ship water samples as well. So if you want to generate and try these samples, we can make the bubbles in the water and they're highly stable, so we can ship them to you and you can try them at your convenience. You don't need to have the unit necessarily. You can just try it, after we've made them for you. You can tell us how much you need. The second part of the question was, sorry. Yeah. Yeah, sorry, sensory. Yes, thank you. Yeah, so sensory side, we have done a lot of work with some quite large beverage companies, and generally what we see can be highly dependent on the type of gas that's in the bubble. So for example, when we fill the bubble with nitrogen gas, we see generally a heavier mouthfeel, a rounder, fuller mouthfeel. We also see some smoothing effects too, where if we have non-nutritive sweeteners or bitter compounds that are in a beverage, for example, we notice that is kind of smoothing off. The edges are kind of removed from some of those beverages too. It's really dose dependent, so it depends on how much you have in there. It could play an effect, but definitely there are those sensory benefits that are noticed, and it really depends on the type of beverage chemistry you have, how impactful it is.
Question. So I know that these bubbles, they're so much smaller. I guess there's two parts. One, does it deliver any sensory changes to the, let's say you're making a beverage and whatnot, and if it, whether it does or doesn't, can you still for example, add carbonate? So you want to carbonate a beverage, can you do them in conjunction and stuff?
Yeah, for sure. So on the sensory side, definitely, we noticed a lot of changes like we just mentioned too, where we do have changes in mouthfeel perception, the weightiness of a beverage, how other flavors are perceived, how sugars are perceived in some cases too. So definitely, we do notice that as well. So yeah, we have seen that. Yes, yeah, exactly.
My question was regarding use of this technology with existing blending systems. Do you know how well they tie into existing blending systems? And if you're creating these nano bubbles, will the mixing and the shear and the recirculation do they have a tendency to destroy these bubbles, and do they lose their efficacy over time?
It's a good question. So definitely, I mean, one of the hallmarks of these bubbles is really their stability. So their lack of buoyancy is a big one, so they do stay around, but they are very tolerant to shear forces. So we can mix them hard, we can do different types of mixing, homogenization people have done, pasteurization is fine with these bubbles. It's really, if we go to a phase change, so if we're gonna go and put these to repeated freeze-thaw cycles for many cycles, or a hard boil for a long time, we'll eventually reduce them. So otherwise, in there, they're quite tolerant, they're quite stable, yeah.
All right, well, thank you, Nick, and a thank you to Hydrosome Lab. They're a bronze sponsor of IFT First, and don't forget to visit their booth at S4603. So before we wrap, quick reminder to please bookmark this session in the IFT First mobile app. So if you bookmark it, you'll receive a push notification that will ask you to rate and share your thoughts from this session. That really helps us to improve the programming year over year. So I encourage you to please bookmark that and share your thoughts. If you're interested in more topics like this, check out Creating Foods for the Future, How Sensory Analysis and Technology Are Transforming Food and Beverage Innovation. And that's presented by Attribute Analytics today at 3 p.m. in the Startup Pavilion. We'll be back here shortly at noon for Unlocking Nature's Color Code in Food and Beverages, sponsored by DSM Firmenich. So come back at noon. Thank you.